Welcome to the Biochemistry Clinic. Today we are looking at the electron transport chain. And at this moment, I think we all know what happens during a glucose breakdown. We know that glucose is broken down to pyruvate, ATP, and NADH in glycolysis. Pyruvate is further converted to acetyl-CoA, which is used in the TCA cycle to produce NADH and FADH2. And we mentioned in our previous lecture that NADH and FADH2 are critical electron carriers. So today we're going to see exactly what happens when the electrons are donated to complex 1 and complex 2. So NADH will donate electrons to complex 1. Now, when complex 1 receives the electrons, it becomes excited. There's energy now. This energy that is now present here will allow the proton pump to pump protons from the matrix of the mitochondria straight into the intermembrane space. And these electrons from complex 1 will actually be directed to ubiquinone or CoQ, which is an electron acceptor as well as a transporter of electrons. Now, FADH2 will come in also at complex 2 to donate electrons. Now, what you should know is that complex 2 is not a proton pump. So the electrons will simply be taken by ubiquinone. So ubiquinone will collect electrons from complex 1 and complex 2. And then, because of its mobility, ubiquinone is going to move the electrons up to complex 3. Now, complex 3 has a proton pump. So it has the ability to pump protons. And we know what happens when electrons come through. The excitement is inevitable. So this becomes supercharged and allows the pumping of protons from the matrix of the mitochondria into the intermembrane space. Once this is achieved, the electrons that are within complex 3 will be channeled to cytochrome C, which is right at the top of complex 3. And cytochrome C is actually also an electron carrier whose job at this moment is to transfer the electrons to complex 4. Complex 4 also gets excited upon the arrival of these electrons, and it initiates the pumping of protons from the matrix of the mitochondria into the intermembrane space. Now, the final electron acceptor is O2, or the oxygen molecule. So from complex 1 all the way through up to complex 4, the final recipient of electrons is O2, which uses the electrons to combine with hydrogen ions and produce water. So this is how the water is produced. So as oxygen accepts the electrons, it forces a reaction between O2 and hydrogen ions to produce water. Now, what you should know so far is that complex 1, 3, and 4 are proton pumps, and complex 2 is not a proton pump. Now, when NADH donates electrons, it becomes NAD+. And when FADH2 donates electrons, it becomes FAD. So you should take note of this. Now, there's something very interesting that you need to know. In the intermembrane space, where we are pumping these protons, you notice that there is an accumulation of protons here. And what do you expect? There will be a concentration gradient. You have more protons in the intermembrane space compared to the matrix of the mitochondria. we know that there's a tendency of substances to move down a concentration gradient. And so these protons are actually now compelled to move back into the matrix of the mitochondria because they're in a higher concentration. But now this movement has to be tactical. So complex 5, or the ATPase pump, will take advantage of this situation because we know that eventually these protons will have to come back. So ATPase pump, or complex 5, provides a channel, it provides a path through which these protons have to pass into the matrix of the mitochondria. Now, the interesting part here is, as electrons move through this ATPase pump, energy is generated by this movement of protons. Now, what is the importance of this energy generated? Well, this is the energy that is used to convert ADP to ATP by combining it with an inorganic phosphate, thereby producing our ultimate molecule, which is ATP. Now, there's something that you have to know. There's a note here. In terms of numbers, for every 4 hydrogen ions that pass through the ATPase pump, you are actually going to produce 1 ATP molecule. So in the next lecture, we shall be looking at the regulation of the electron transport chain as well as the inhibitors of the complexes. But before that, there's something that I would want you to know. Now, these proton pumps, proton pump 1 and this one in complex 3 and the other in complex 4 will pump protons into the intermembrane space. But the question is, how many protons are pumped by each of these proton pumps per mole of NADH and FADH2? So for every mole of NADH and FADH2, this proton pump is able to pump 4 protons into the intermembrane space. This one is able to pump 4 protons into the intermembrane space, and this one is able to pump 2 protons into the intermembrane space. Now, initially, there's supposed to be 4 protons that are able to be pumped from here, but the other 2 protons are the ones that are actually used to combine with molecular oxygen to form water. That is why complex 4 is only able to send 2 protons into the intermembrane space. So if you do your maths well, you're going to realize that the 4 in complex 1, 4 in complex 3, and 2 in complex 4, will form a total number of 10 protons that are pumped into the intermembrane space. Now, this number will be very important in our next calculations in the next lecture, where we're going to now try and calculate or try to find out how we arrived at the conversion that 1 mole of NADH is equivalent to 2.5 ATP molecules, and also 1 mole of FADH2 is equivalent to 1.5 ATP molecules. So those of you that know your mathematics very well, you can already do the math now, because you have this conversion that for every 4 protons that pass through the ATPase pump, you produce 1 ATP. So you can actually prove now why we say that 1 NADH is equal to 2.5 ATP and 1 FADH2 is equal to 1.5. So do not miss the next lecture. Thank you.